Everything's going fine. Just a few slides on aging research, again from the in silico perspective. And you've basically seen that slide already, I think, 2 days ago by Olshansky. And that's I just want to present that as kind of really graphical motivation to do aging research. And this is about that woman and its life expectancy in the United States, 1990, if certain diseases are eliminated. And life expectancy would change only slightly if you can eliminate entire classes of diseases, maybe up to 16 years if all cardiovascular diseases, diabetes and cancer are eliminated. But then of course there may be other diseases like Alzheimer. So this is a strong motivation for me to work on the roots of the problem, namely on aging research. And I'm not running for some time workshops, especially there have been 2 workshops on systems biology and bioinformatics in aging research. And we published a report of the first workshop, and this is the one of the second workshop. And you see that quite a lot of people have worked on that, and it was writing this report was based on Google Docs, on a wiki-like approach, where we all together as a group formulated then some recommendations for aging research. So this is published in Rejuvenation Research, and you can refer to it. And basically, the workshop is displayed in a graphical way on this slide. So basically, the topics have been stem cells, epigenetics, diet, growth, DNA damage and cancer, inflammation, and Holz and mitochondria. And from the bioinformatics perspective, gene ontology was very important, modeling was very important, genome-wide association studies, and network biology, next generation sequencing, and screening support. And we suggested some actions in the conclusion of the report. In particular, we proposed to build a wiki-based collaborative resource that provides data integration on all levels, because we believe that is really tremendously important for aging research. As Ovid Gray is also doing similar literature scannings, but they should be integrated into a large database and made interoperable and computable. And I think this is such a huge project that it can only be started by crowdsourcing effort, in a way like Wikipedia was done. And so this one of course would be a much more scientific project, but I believe it is now the right time to start crowdsourcing such an effort. And there are some people who have started that, some of you know Georg Fuellen's aging map, which is actually also on the here in this room displayed. And there's a human aging diagram I just learned from the previous speaker, the Russian initiative. There's also Langley's interactive environment for explanatory biological modeling, which he also presented in Rostock. And in particular, there's an enthusiastic graduate student, Daniel Wuttke, who's working in the lab of João Pedro de Maireles, and he's working on what he calls the Enigma. This is a project based on the Django software, and this is a starting point for crowdsourcing such a compilation of aging-related knowledge as supported by ontologies, and the human and machine, so using human curation and also machine learning and electronic annotation of data, data unification, integration, and also constructing networks and supporting crowdsourcing. Maybe with this Django website, everybody can join in. And I would propose that Daniel Wuttke is really one of the people who is best poised and qualified to be one of the drivers of such a project. Together with some other people, there's also at this conference probably coming today somebody from Munich who's working in Munich and in Cologne, also working on DNA methylation for sense-related projects. So there's, I think it will be the mass now to get such a project going, and maybe this presentation, this conference is the starting point for this. Yeah, and our second action item actually was, since this was also a bioinformatics conference, that molecular agent research should be advertised as the one prime application area to the main bioinformatics stakeholders, namely in particular to the European Bioinformatics Institute, and of course also to the National Center for Biotechnology Information, the NCBI in the US, and to similar institutions worldwide. So we believe that aging has a lot to do with many biological processes, but those are the biological processes we care most about, and are the ones that are most relevant for aging. And so they should be prime application area for bioinformatics initiatives. And finally, we had a third action item, which is more like general considerations for aging research, focusing more on early life interventions, and on the other hand on aged animal cohorts, covering an area of species, not just like 6 mice. And also starting with intervention screening, and yesterday I presented some bioinformatics support for intervention screening, which I'm really keen to continue. And just one slide, in Rostock we've got a very nice role model for interdisciplinary efforts. That's the Department Ages, Aging of Individuals and Society. This is part of another unique Rostock institution, the Faculty of Interdisciplinary Research, or School of Interdisciplinary Research. They're doing dementia research, Alzheimer's, assisted living, longevity, demographic change, and molecular aging research. And I think this could also be a nice thing to be copied by other universities or similar institutions. Yeah, and lastly, again, you've seen that slide before, but maybe not all of you. I'm inviting you to contribute to 2 workshops. One is on aging research, which will be the 20th to the 21st of September 2013. There will be ontologists speaking, like Paul Scofield, Udo Hahn, Friedrich Schumann. There will be Jean-Pedro de Meinderhes. Probably Ulrich de Brey will be there, with I think 80% probability is that. And on Sunday, the 22nd, we will actually have a kind of a Sunday brunch on more philosophical topics, where Ulrich will probably be speaking and Jean-Pedro. And this will be in a very nice place, Osterwaldemünde, which is really a nice holiday resort at that time. And this is supported by the aging department at Rostock. And this will also probably be most likely sponsored by SENS, and previous events have been sponsored also by pharmaceutical companies and by the ministry. And since I've been talking yesterday on stem cells, there will also be a stem cell bioinformatics workshop. Also one of my regular activities, there will be very prominent speakers from the US, and that will be in Cologne in April 22, 2013. And later on the same week, there will be an entire conference on stem cells, the NRW Stem Cell Network International Meeting. Yeah, and that's it. What I could do, if you are interested, I could present my own Rostock aging project, just 3 slides. But maybe just, this is a separate topic, we should maybe start discussing this first. If you've got any questions, please ask. And thank you for your attention so far. Questions?
Do you think any similar projects can be initiated in other places, in other countries? I mean, there's no comparable project anywhere else.
What do you mean similar to what? To the Enigma crowdsourcing, or to those conferences, or to the
crowdsourcing.
I think it must be international. This can only be done in an international way.
No, I mean for other projects, there was talk, for example, to do crowdsourcing for mice and crowdsourcing. Maybe I just didn't get the point. I'm sorry.
Okay, for mice, you mean for mouse research?
Yeah.
Yeah, I'm not aware of any people who are kind of creating a big mouse resource and a crowdsourcing effort, but I imagine that maybe, I'm not aware of that. Other questions? No? Okay. Should I spend 3 minutes? Yes, 3 minutes? Okay, just a few slides. Okay, so there's a brief introduction to an aging project I'm running in Rostock. It was also with systems biologists, and this is a collaboration also with Sally Ibrahim from Lübeck. It's funded by the German ministry as part of the Gerontosis initiative. And you've seen in the previous card this Gene Age Center, which is also funded by Gerontosis, which is part of a very large aging-related effort in Jena, where they also have a large institute on aging and aging-related diseases. So that's funded by the ministry. That's Sally Ibrahim in Lübeck. He has developed mouse models, and they are very special. They are not like the mutator mice, where you have random mutations in the mitochondrial genome, but here you have very specific mutations. Those are contrasting strains. They have got one single mutation in all the mitochondria of the mice, and that's done by a strict breeding scheme. And they are affecting all the mitochondrially encoded electron transport chain components, complex 1, 3, 4, and 5, if I'm correct. And those mice are now bred. We've got hundreds, actually thousands of mice now, which are then followed up along their lifespan from 0, 3, 6, 12, 18, until 24 months. We take data at certain time points, response to a stimulus, to a stress, and then we are measuring aging-related molecular pathways, cell and organ functions, and lifespan. And it's altogether, it's 10 groups in Rostock, Lübeck, and Leipzig. And those data that we are obtaining from the mice, they are also used for systems biology modeling. And this is a basic setup again, and a kind of visualization that we have got the mice that we are following up across their lifetime. We've got those specific mutations that ultimately we will investigate up to 10 to 15 various mutations of electron transport chain proteins and other mitochondrial encoded genes. And we are measuring the data in several organ systems, like central nervous system, immune system, pancreas, and fibroblasts. And the data are mostly protein data, because we are doing blood data of the various kinases and NF-kappa B and the complexes, beta-galactosidase and other senescence markers. We are measuring ROS and ATP. And that means we've got a lot of input in experimental data. And then we would actually like to do a machine learning effort in trying to take those data and trying to learn organ vitality, functional data from the molecular data. And also trying to learn life expectancy of the mice. This can of course only be validated once the project is over, once the mice are up to 3 years old and once the mice have all died. So we will also use systems biology modeling to predict parameters and mechanisms, the signaling up-down that I've been talking about yesterday. And we'll integrate the data of oncologies to similarity measurement, clustering, machine learning. And I would like to know which kind of mutations at which point of life in the mouse, in which organ system has the strongest effect on aging. It means it is kind of very important also to look at various aging theories like hyperfunction versus damage, for example, and especially looking in particular at the role of ROS. And very interesting would also be a prediction from data of 3 to 12 or 12 months to the 18-month data. So is there any structure, any underlying molecular mechanism that can be found in that data that is driving the aging processes? So we've got models of the respiratory chain, ROS production, DNA damage, P53 inflammation, related signaling, and cell aging and dysfunction. And those are some of the interconnections of the things that we are measuring, like P53, P38, junction DNA, and some others. And this is supposed to yield a small systems biology based model. Yes. Thank you very much. Questions?
I didn't know about these mice. Can you tell me any more about them? I mean, since these mutations are not lethal, I guess they must be, if they are present in every copy of the mitochondrial DNA in
Yes.
the mice, then they must be mild.
Right. There's mild impairments,
Okay.
of course.
How were they created, do you know?
It's published, I think, 2009 in Genome Research by Sally Eberlin, and this is a strict breeding scheme. So it's, he has been breeding those mice for 10 years, as far as I know, and we are also regularly checking that it's really almost all of the mitochondria are still harboring the same mutation.
Okay.
Because that's important for the model. It's just the opposite of this random mice that Larsen, for example, and Trifanovic are looking at.
Okay, thank you.
Other questions?